Hello, everybody. My name is Gadi Taubenfeld, and I will speak about a joint work with Ziv Bar Yosef and Sabrina Rashid from CMU. So the title is The Epigenetic Consensus Problem. So here are 3 facts about the human genome. It is 2 meters long. It has 3 billion base pairs, and it includes about 25,000 genes in it. So how do you take 2 meters long genome and condense it so that it will fit into the nucleus of a single cell? So here is a kind of an idea. So you start with the double helix DNA. This is the double helix DNA. You wrap it around proteins which are usually called histones. Okay, so here is the DNA. It is wrapped around this histone, and then around this histone, and so on. Okay, then you take all this structure and condense it even more. This is what you see in this picture. This is called the chromatin fiber. And then you take the chromatin fiber, condense it even more, and it fits into a single cell. So we will get back to it in a few seconds. Now here is the question. You have 2 different cells in here. So how can an organism have 2 different cells types, yet one genome? So those 2 cells, or all the 200 cells, different type of cells in our genome, in our body, has exactly the same genome, exactly the same DNA. So how come a neuron, which has exactly the same DNA as a skin, become a different thing? And the answer obviously is that if you look at the genes inside the genome, then each cell express or turn on only a fraction of its genes. The rest of the genes are repressed or turned on. So the genes that are on in the neuron are different from the genes that are on in skin cells. And we need this background in order to understand what is going on later on. Okay, so the next question is how we turn genes on and off. Okay, we said gene can be either on or off. Okay, so look at this picture. Here we have condensed chromatin. Okay, so this is the DNA and it wraps around these histones that we said. If it is condensed, and let's assume that this is the red one is a gene. So if it is condensed and the gene is not accessible, it's not possible to get to it, then we would say that the gene is off. If it is in this picture, the gene is accessible so that somebody can get to it, replicate it, use it in some way, then the gene is open. So the question is, how do we get from this situation, how do we activate a gene? So how do we get from this situation into this situation? And backwards, how we can take a gene which is on and turn it off, deactivate it. Here's a picture of the same thing. So here we have the chromatin. This is the DNA, as you see in here. This is a gene that is accessible, so it is on. Actually, it's a necessary condition. It's not a sufficient condition. There is also something which is called DNA methylation, but we're going to ignore it. So here is the gene which is on, okay? Here is the DNA again, it is wrapped around those histones. And here we have a gene, the red one, which is off because it's not accessible, okay? And you should know that genes are turning on and off all the time in our body. It's not only in our childhood that you decide what cell would become, and then the genes are fixed either on or off. As a result of a diet or stress or pollution, the genes in our body are changing state from on and off, backwards all the time. Okay? So how this happens? Let's look at this picture, okay? So what we have here, we have, again, the DNA. It is wrapped around the histone. So this whole thing, this histone plus the DNA that is wrapped around it is called nucleosome. Okay, and here we have this whole chromatin. Some of it is, part of it is condensed and part of it is open. Okay. Now, if we look at the histones here, they have what is called histone tails. Those are tails, and there are proteins that are floating around. We will speak about them in a minute. They can actually leave marks on the histone tails. So in some way, you can think about this nucleosome as a memory location, and those proteins that leave marks are actually writing into this memory location. Okay? So we have this memory location here. This is how we actually model the biological system. And we have three types of general types of proteins. They are called writers, erasers, and readers. Okay, and those are names that are given by biologists. It's not something that we are inventing. Okay, so writers can actually leave marks on the histone tails. Erasers can come and remove marks from the histone tails. And readers actually observe what is the state. They are the executors, and we are not going to focus on them. We're only going to focus on writer on erasers, because the synchronization problems that we are going to introduce, or that relates to how genes are activated and deactivated, involves only writers and erasers. Okay, so we have three types of them. And here is another abstraction. From now on, everything is new. This is what I just told you, what was a background. So we're going to abstract it away, and instead of speaking about lots of types of writers, we're going to say that there are two types of writers. A one writer, which can actually leave a mark, which we called one. This is corresponding to writing one into this memory location. And a zero writer that can actually write zero into this memory location. It simply leaves a different type of marks. And then we have two different proteins, which we called zero eraser and one eraser. The zero eraser can come and erase zero marks, and the one eraser can erase one marks. Okay, so let's assume that this nucleosome or memory location, the state is currently zero. There is a zero mark. So if a one writer comes, it cannot simply write one on top of the zero. The way that it has to go, and this comes from biology, we are not inventing anything here, we're just modeling the biology assumptions. The zero eraser has to come first. It has to remove the zero mark that is somewhere in here. It has to remove the mark. Now we have the cell is empty. Only now the one writer can come and write one, leave its own mark. Okay, so this is how it works in biology. Now, the thing is the following. We have those proteins that from now on we're going to call them processes. So we have four different kinds of processes, as we just said. We have this memory location in here, the nucleosome. That is how we model it. And they're floating around, and at some point it might be that this zero writer can write zero here, and another zero writer can write zero here, and then the one writer can write one in here, the other one writer can write one in here. Okay, and we have a mess. Part of the memory locations, the nucleosomes, are zero, and part of them are one. Now here is the most important information about the biology part, how we turn genes on and off. In order to turn off those genes that reside over here, this part is open, so if there are genes in here, they are accessible, so they are on. What we have to do is we have to get into a situation in which all the memory location, all the nucleosomes are written zero. And then by magic, this part will be condensed. Similarly, if we want to open up this part over here, we want to expose some genes, so we want to activate them, then we have to write one in all the memory locations. And then by magic, this part will open up. And the synchronization problem is how come that all the locations that when the cell wants to deactivate some gene, let's say a gene that lives in here, all these memory locations are written by zero. How this happened? saying differently, what is the algorithm that each one of this type of processes or proteins are running so that this is enabled? Okay, here's a different way to think about the same thing. So now we are using only distributed computer terminology. What we have an array of cells. So this is those nucleosome. Each nucleosome corresponds to one of those locations, and there are many of them. So the cell is huge. There are hundreds of thousands locations or cells in this array. And we have two teams. One team is the team which is called 1, which includes the one writer that tries to write 1 in here, and the other one is the 0 erasers that are helping the 1 writers. They are going to erase zeros to make space, to make room for the 1 writers to write 1. So this is one team, and the other team is the 0 writer that tries to write 0 here, and the 1 erasers that are helping them. They are erasing the ones. And the problem, the epigenetic consensus problem, is to find the algorithms that each one of those 4 types of processes are running, so that with high probability, the bigger group wins. So what do I mean by bigger? If one group is, say, the ratio between the size of the group, the number of participants here, if the ratio is, say, 1 to 3, so if the one group is 3 times bigger than the 0 group, then we want to end up with high probability that eventually this whole array is written 1, and then the algorithm stabilizes. This array would not change from that point on. And this will cause this part, for example, to open up. Okay, so this is the epigenetic consensus problem. Okay, so there are 2 interesting parts in here. One is how we model things, and one is the algorithm. So let's speak about the modeling, because we are using a very weak model. So we are going to allow randomization. In biological algorithms, you always use some kind of randomization. Note that the processes are anonymous. That is, there are no IDs for the processes. Those little proteins, they have no IDs. More interestingly, the memory is also anonymous. That is, there's no agreement between the processes of the names of the memory locations. So this is a new area of research in the last 4 years, and I encourage you all to look at some papers, both in Sirocco and in Pozzi, and to read about this. I find it very interesting. The next thing is the processes should be memory-less. This comes from biology. Well, we're going to assume that they have 1 or 2 bits of memory just to model things that has to do with randomization and the direction that they are moving, but not more than that. But in reality, they should be memory-less completely. As we said before, transition from 0 to 1 cannot occur directly. The eraser can come and has to erase before a new value is written. There's no sense of direction. There's no left and right for the processes. The proteins do not agree on the left side and the right side, or the right side of the array. And we would like the algorithm to be self-stabilizing. So actually, the algorithms that I'm going to mention, with a tiny change that we describe in the paper, you can make it self-stabilizing. Okay, so this is a very weak model, and this is why one of the interesting part in this paper, to my opinion. So what we do in the paper, we present an algorithm that matches the biological assumptions, prove its correctness, and derive bounds on its expected runtime, both theoretically and in simulations. Okay, so let me give you an idea how the algorithm works, and it is very simple. So in general, distributed biological algorithms or algorithms that run in nature are always simple, almost always simple. So this is a very simple algorithm. I should say that it took us some time to get to a simple algorithm. Usually you start with a complex algorithm and then you simplify it until you get to the right thing. So the fact that it's simple, the algorithm is simple, it doesn't mean it was simple to find it. So here is the algorithm. If we look at the one-writers here, this is the writers, right? So if we look at the writers, the code for the writers is very simple. The one writer simply scans the array back and forth. So this is what the writer is doing. Whenever a writer is seeing an empty location, it writes one and continues. So it just moves back and forth. And similarly, the zero writers also scan the array back and forth. And whenever a zero writer sees an empty location, it writes 0. The algorithm that the erasers are executing is slightly more complicated. What the erasers, both the zero eraser and the one eraser, are looking for in the array are what we call the collision. This is a collision. A collision is a pattern of 0, 1 or 1, 0. Okay, two adjacent locations which has different values. So whenever an eraser, say a one eraser, so this is a zero eraser. So whenever a zero eraser finds such a collision, it simply erases, in this case, the 0. And if the one eraser here, if the one eraser finds it, it erases the 1. Okay? So the eraser again scan the array, looking for collisions. Whenever they find collisions, they resolve the collision by erasing one of the location, depending whether they are 0 eraser or 1 eraser. And we have to prove that this actually stabilizes and the bigger groups with high probability will eventually win. Some intuition why the algorithm stops, this is not an analysis, but just an idea, is that whenever some location is erased, the probability that the value of the bigger group will be written there is higher. Okay, so over time, it will converge to the values that the bigger group is trying to write. And obviously, once it converges, once all the values are the same, then the erasers would not be able to erase anything, and it's stabilized on this value. Okay, so this is one of the algorithms, or the basic algorithms, the main algorithms that we have in the paper. We have variants of these algorithms also that we discuss. For example, it can be that that the writer, whenever it scans and see a collision, simply waits on this collision until one of the values is erased, and then it tries to write into it. This is another version which by the simulation shows that it's faster than the one that I described before, but it's much harder to analyze. So we analyzed the first version that I mentioned, that the writer simply scan back and forth, never stop, and whenever they see an empty location, they write the value that they're supposed to write. Okay, so in the analysis, we did the analysis of the probability of reaching agreement. And this is a corollary of the more general result that we have in the paper. So for example, if we look at the ratio of 1 to 3, so let's assume that the bigger group, this simply said that the bigger group, say this one, the one group, is 3 times bigger than the other group or more, then the probability that the final decision value is 1 is more than this expression where n is simply the size of the array. Okay, so we get what we want here. And then we also computed the expected number of steps that it takes until we converge. So again, in this case, one group is bigger, then we have the expected number of steps until we converge as a function of the size of the array. Okay, and the analysis is not too difficult. What it uses is it uses results from random walks in one dimension and a simple, relatively simple application of Chernoff bound, and we get this. Okay. In addition, we have simulations, and the simulation actually verify what we showed you in here. This and we checked also the simulation. Also, we simulated various variants of this algorithm, as we said. Also, when the writers are spinning on the collision, waiting there instead of just moving back and forth, and we compare this the various variants to the main variant that I just described, for which we have this analysis. There are many more results in many more information in the paper, much more information about biology, and some evidence that the algorithms that we are showing you here, which is based on finding collisions, is actually the one that runs in reality. Okay, thank you very much. This is it.